So, ladies and gentlemen, welcome to today's webinar sponsored by Spectradyne. We're delighted to welcome Dr. Jean-Luc Fréquin, who is the CEO of Spectradyne, to deliver the talk today. Dr. Fréquin is a very engaging presenter and always gives a very exciting talk with lots of questions. So, ladies and gentlemen, if you have questions, you can type them in the Q&A box over the course of the presentation. Alternatively, you can raise your hand at the end of the presentation section of the webinar. We'll then unmute you, and then you can ask your question in real time to Dr. Fréquin and have a discussion, as much as almost like a physical conference where you have a back and forth discussion with the speaker. So today's topic is very exciting. Spectradyne has been leading the way with instrumentation and technologies in particle analysis, and they will be giving the talk today discussing extracellular vesicles, lipid nanoparticles, and the technologies and instrumentation that they have as a means to interrogate these particles and study their biology. So without further ado, ladies and gentlemen, we'd like to welcome Dr. Jean-Luc Fréquin to deliver the webinar presentation, and we look forward to a very engaging webinar. Thank you, and welcome, Jean-Luc.
Thanks very much, Inal, and welcome to everyone attending today, and thanks for your time for joining us. In this webinar, we'll ask the question, how is that black box really measuring my lipid nanoparticles or extracellular vesicles? And we'll do that by investigating in detail how the most common techniques work and what kind of information they deliver about the samples you're trying to measure. I hope that you'll come away from this with a deeper understanding of the measurement techniques you're using, and ultimately that your science will be better, faster, and easier, because that's what we all want as scientists, right? My goal for the webinar is to encourage you to think about three things. First, that you should take particle size and concentration measurements seriously because they're critical experimental parameters, and controlling them better can help your science. Second, I'd like for you to be asking yourself if your measurement technique is doing a good job of delivering the information you're asking it to. And so we'll investigate the limitations of different techniques to get at that question. Then finally, I hope the examples I'll show today will convince you that using multiple measurement techniques can provide key insights for your research that you can't get from any single method on its own. We'll start with a short introduction in which I'll make the case for why particle size and concentration are so important, give a brief overview of common nanoparticle measurement techniques, and then provide a quick review of Spectradyne's technology so we're all on the same page about how it works. Then we'll talk in more detail about three of these techniques: dynamic light scattering, nanoparticle tracking analysis, flow cytometry. And for each of these, I'll show specific examples to illustrate what we've talked about in terms of how they work. So let's get started. Particle size is important essentially because it impacts the particle activity or power. Let me explain what I mean by that. Particle size determines the diffusion rate of the particle in a system, and so how quickly it can reach a target in a drug delivery situation, for example, and also impacts the clearance rates of these particles from a biological system, right? So as such, the particle size determines the bioavailability of a drug, as an example, and ultimately it has a significant impact on the drug efficacy, how well it performs the task we're asking it to do when it's administered. Particle size also determines the particle volume, right? That's kind of obvious, but when you think about it, the bigger the particle is, the more you can fit into it. So that's the particle, the payload volume. And that's a critical experimental parameter because it's essentially the per particle dose of whatever you're trying to deliver through these particles. So size is definitely a critical parameter and ultimately impacts, in a therapeutic setting, the safety and the efficacy of a drug being encapsulated by a particle. Concentration is also important, essentially because it represents the overall dose of whatever you're trying to deliver, or the amount of active ingredient you're using in an experiment. An obvious example that's present in our minds right now is the mRNA vaccines that are lipid nanoparticle based therapeutics. And in this case, the number of particles per mil, in conjunction with the size of these particles, really is the dose of the drug, right? So it's super important to measure this accurately, and it must be carefully controlled, for the safety and efficacy, again, of these products. So those might seem kind of obvious statements. Let's take it, let me illustrate that with an experimental example. So why are these both critical parameters to measure accurately? Well, suppose you've prepared two different formulations, A and B, and you want to compare the efficacy or the biological activity of these different samples. And they might be a therapeutic, or they might be isolated EVs that you're interested in understanding their biological activity in a system. So first step is you prepare the two different samples, and then you apply them to a test system. Maybe it's maybe it's an animal model, or maybe it's cell culture or some other system. And then we measure the outcomes of the experiment in a quantitative way, right? I think no scientist on this phone call would do this experiment without first controlling for the amount of material, A and B, that you put into the test system, right? And so hopefully this comes at this from the experimental side of thinking about why these things are so important. Accurate size and concentration are critical metrics for good science, critical parameters that need to be controlled to do good science. So how are people quantifying nanoparticles currently? So let's go through a few of the most common ones. Dynamic light scattering, DLS, is still very popular in the nanomedicine space, particularly for LNP formulation scientists. This technique is an optical technique, like almost all the existing ones. And in particular for dynamic light scattering, it's ensemble averaging technique. So we're not measuring single particles with it. And we'll talk about how that has benefits and drawbacks later. One of the technologies that's very popular in EV space, and really is a foundational technology for the whole field, honestly, is nanoparticle tracking analysis. This is another optical method. It uses similar principles to dynamic light scattering, but in this case, it's a single particle measurement technique, and that comes with certain advantages also. And more recently, there has been efforts to include flow cytometry as a technique for measuring nanoscale particles. And so we'll talk about this one a little bit later too. There's another optical detection technique. It's single particle. We're counting particles one by one. And it's emerging as, I think, a pretty promising avenue for nanoparticle analysis going forward. You'll notice that these three top measurement techniques, though, they're all optical methods, right? And so they're looking with a very similar, or sometimes the same, lens at what's in the sample. So let's look to what we call orthogonal techniques, and that's not our term, but it means that a way of analyzing particles with a fundamentally different approach, right? So tunable resistive pulse sensing, this is an electrical method. It's been around for a while. We're a fan of the approach, the electrical approach. This particular implementation is challenging to try to use it. But that's where Spectradyne's technology comes in with Microfluidic Resistive Pulse Sensing, or MRPS. This uses, again, electrical detection. It's based on the Coulter principle, which has been a gold standard for decades, counting large particles in cells. And so it's a non-optical technique. So it truly is orthogonal to all these top three optical techniques. It's a single particle measurement method, and it's fast and easy to use. Let me give a brief overview of this technology so we're on the same page about how it works. This is a picture of our product, the NCS1. It's a benchtop instrument. It's pretty small footprint. It's about a foot wide. It measures concentration size accurately, starting at about 50 nanometer particle diameter and all the way up to 10 microns diameter. As I mentioned, it uses electrical detection, so it's a non-optical technique. And it's pretty fast. Generally, it takes 5 or so minutes to measure a sample. One of the features that our customers really like is that it uses microfluidics to deliver its performance. And one benefit of that is that only a very small sample volume is required for analysis, as little as 3 microliters. So let's talk about how it works so we can compare it to the other technologies a little bit later. So microfluidic resistive pulse sensing, as I mentioned, is based on the Coulter principle. particles are analyzed one by one, and the concentration and size are measured in about the most direct way that you can currently on the market. So the way this works is we flow the sample through a very small opening, an aperture or a constriction. And every time a single particle passes through this constriction, we get an electrical signal. We measure two things about this electrical signal. One is how big the signal is, because that tells us how big the particle is, tells us the particle size. And we also measure how fast it goes through the constriction, because that tells us how much of the sample has flowed through here. So that's the volume of the sample that's been analyzed. So this is real data from a single particle event. You can see how quickly things are moving through. If we measure for longer, you get data that looks like this, where I've highlighted every particle event by a red dot. And so here, right away, you can see there are multiple sizes of particles, right? There are 4 different bands of peak amplitudes. And we can see that just by looking at the histogram of the counts in the sample. But because we were measuring the volume of the sample that we measure that we've analyzed, we can do better than just counts in a histogram. We can convert these counts to actual concentration, absolute concentration numbers. And that's what this is showing you here, is the final product of the analysis of this data, which happens essentially in real time while you're measuring. This plot is showing nanoparticle concentration on the y-axis and particle diameter on the x-axis. And you can see those 4 populations are readily resolved. This sample was a mixture of polystyrene beads, 50, 90, 120, and 150 nanometers in diameter. You can see all the different components of this mixture were detected, and resolved individually. And we can be quantitative on this plot over any size range that we want. So maybe you're not interested in the particles themselves, but in the byproducts or something that's happening in between here, you can directly measure the concentration here over any subrange. This combination of accurate size and concentration with the ease of use of our platform are not currently available with any other method. Okay, so we've now reviewed, high level what is available for technology commonly used for nanoparticle analysis. Let's dig in more deeply, starting with dynamic light scattering. So this technology, as I mentioned, in our experience in the field, is still used by the vast majority of nanomedicine scientists. And so it's important to understand how it's working and what kind of information it's okay to extract from this. So dynamic light scattering is an optical detection, and it's ensemble averaging. So how does it work? Well, you start by shining, essentially illuminating the sample with a laser. And we're doing this for many particles at once. So all the particles in the laser beam are being illuminated. And then we monitor the scattered light that comes from these particles. Now, the particles are not standing still in the solution, obviously. They're undergoing Brownian motion. And so by monitoring the dynamics of this scattered light, so how that scattered light changes over time, we can infer the Brownian motion properties of these particles in the sample. Remember, the signal is coming from all the particles at once. And so this is really like an average measurement. So from this Brownian motion information, we can infer the average scattering size, essentially, of particles in the sample. However, you'll know one thing is, first of all, we're not counting any particles, right? And the intensity of the light that's coming off here is not telling us about the number of particles in the sample. So DLS doesn't deliver any concentration information, actually, right? So we're already missing the dose component of this. Now, it's a little bit more subtle too, looking at the scattering intensity. So particles scatter light depending on how big they are. And the intensity of the scattered light depends really strongly on the diameter of the particle. It scales with the diameter to the 6th power. So that means if we say a 50 nanometer particle in the sample scatters 1 unit, then a particle that's twice as big scatters 64 times as much light. And a particle that is twice as big as that scatters 4,000 times more light back to the detector. And so as a result, when we look at polydispersed samples, dynamic light scattering has a real problem with that. Large particles in the sample scatter so much more light that they dominate the measurement result. And we'll see examples of that in a bit. So let's look at some real data. So that's what I'm showing here. This is a DLS measurement of a real sample. And if you are a scientist looking at this, you're, you might be thinking, well, this looks pretty promising, right? I've got a nice clear peak. The z-average measurement from DLS is saying 165 nanometers. It looks like, it's a nice bell-shaped curve of distribution, right? However, you're a good scientist, you're careful, and so you remember that even the ASTM standard for using dynamic light scattering says that this distribution it's an intensity distribution, and it's not a particle size distribution. It's not delivering any concentration information. In fact, the ASTM standard goes on to say that conversion to a particle size distribution is ill-advised and fraught with danger. And those are ominous words, but the reason is because it's a mathematically ill-posed problem, essentially. What do we really know about this sample? Well, what we really know is the average diameter of scattering, the effective scattering average diameter of 165 nanometers. Why is that? Well, a sample that we would love to see generate a scattering distribution like this would have a normal distribution centered at that diameter, and it'd be nicely well-defined with a narrow distribution, right? This would be the dream. You've got a nice, well-formed product, right? However, you can get the same z-average from a much lower concentration version of the same thing. Remember, we're not getting any concentration information from this measurement. And you could also get this z-average from a much more complicated particle size distribution like this one. So here we have a lot of small particles, but they scatter less strongly than these fewer big particles. And on average, they generate something in between somewhere, right? Like 165. So really, dynamic light scattering is telling us neither the particle size nor the concentration of particles in the sample. And what we need to get at, what's really going on here, is an orthogonal measurement technique. So at Spectradyne, we measured the sample on our NCS1. And this is what the actual constitution of the sample is. It's, again, a mixture of polystyrene beads. These are 50 and 94, 122, and 150 nanometer beads that we got commercially mixed all together, right? About 2 × 10⁻⁹ particles per mil each. And you can see exactly what we talked about here. Dynamic light scattering is picking up mostly the big particles. And so the DLS Z average is somewhere around the big particle population in the sample. And it's really not telling us actually what's going on in the sample. The NCS1 MRPS, in contrast, gives the high resolution sizing that you're looking for to actually know what's in the sample and can deliver the absolute concentration on any size range. And so this is what we learned by looking with a fundamentally orthogonal measurement technique, right? Not looking at it just with dynamic light scattering. Now, this is a bit of an unfair comparison, maybe you'd say, because dynamic light scattering is an ensemble method and MRPS is measuring single particles, so it has an inherent advantage, and you'd be right. But the fact is, many people are still using DLS to measure highly polydispersed samples, and it's failing them, honestly. So let's look at a specific example. So this is an example of lipid nanoparticle formulations made by 3 slightly different recipes. Okay, and these were first measured by dynamic light scattering. So sample 1 was measured with a Z average of 66.5 nanometers with a polydispersity index that's pretty reasonable. And sample 2 was measured with essentially the same Z average and slightly higher polydispersity index. Sample 3 was unable to be measured by DLS, presumably because it was considered too polydispersed by the sample or by the DLS instrument to be appropriate. So then we took these samples. So this is all the information we get from DLS, right? And the end users that were looking at these samples were stumped because there were different recipes. They expected to see changes, but they couldn't measure them by dynamic light scattering. Then we measured these samples by the NCS1. And these are the particle size distributions that we obtain when we measure them using MRPS. So sample 1, you can see well, so right away, there are clear differences between the three different samples, right? They have peak mode diameters that vary between the three different samples. And they have rich distributions, right? Green sample 2 has sort of a bimodal hump on the side here, and sample 1 has larger, a higher concentration of larger particles here above 100 nanometers. And so there's a lot of information in these particle size distributions that you just can't get from DLS on its own. And this highlights the importance of using orthogonal techniques. So this just to remind you that dynamic light scattering averages for samples 1 and 2 were the same. They couldn't distinguish between the two. And even this Z average is not really that relevant to the size of the actual particle size distribution, right? All right. Not only that, for the size distribution, MRPS is delivering the concentration over any size range of interest under here and counting tens of thousands of particles in a few minutes to do that. Okay, so to summarize dynamic light scattering, it always gives an average particle size, which is handy and works very well for monodispersed samples. That's great. However, I think I've shown to you that for polydispersed or unknown samples where you don't know a priori what you're going to be measuring, that dynamic light scattering really fails to give much useful information at all. And that what we need is an orthogonal technique to measure the actual size and concentration. Okay, I saw a question in the chat about the difference between NCS1 and NTA. So we're about to talk about NTA, so hopefully this will answer your question. So let's talk about NTA and how it works and its benefits also. So in NTA, nanoparticle tracking analysis is very similar to DLS in the sort of fundamentals. So the particles are undergoing Brownian motion. So we illuminate the sample with the laser. Particles are undergoing Brownian motion, except now instead of detecting all the scattered light, what we'll do is we'll take a video of all the particles doing their random walks with Brownian motion and track each individual particle track. So we're still detecting scattered light, but now we're detecting scattered light from single particles, and we're watching them go around do the Brownian motion walk. And from the radius of the radius of these random walks, we can infer particle size of each one, and because there's a dependence on how much space these explore to the diameter. So that's good, but there are similar limitations here because we're looking at scattered light. So again, the scattering intensity is proportional very strongly to the diameter of the particle, to the 6th power of the diameter. And so what that means is that in these videos, the smallest particles are very, very dim relative to the larger particles in the sample. And ultimately, in real-world samples, the large particles are too bright for the camera relative to the small ones, and we miss the small particles. And I'll show you an example of that in a second. So let's look at an illustrative example where we look with polystyrene beads. Okay, and this is an experiment that you can do quite readily yourself if you have an NTA and polystyrene beads on hand. So what we're doing here is we're measuring a monodispersed sample of 208 nanometer polystyrene beads. Okay, and you can see NTA measures it just fine. The diameter's right. Concentration we've normalized here, but it comes out as expected. We can do the same thing for a monodispersed 150 nanometer particle and get the same result, right? We get a nice well-defined peak here, and the concentration is coming out about expected. Same thing for 94 nanometers, and you could continue this down as long as the particles are scattering strongly enough down to a few tens of nanometers even for gold particles, for example. So from this experiment, we might suppose that the sensitivity of NTA of the NTA method should be less than 94 nanometers diameter, right? So what gets complicated is, or what complicates that conclusion is when we start looking at a polydispersed sample. So if we mix these 3 particles together, we would expect to have all 3 particle populations in the sample. And but when we do that and measure it on NTA, this is what we get. Okay, and what you can see is that the 94 nanometer particles, the smallest ones, are not being detected when they're in the mixture. What we've shown essentially is that the sensitivity limit of this technique depends on the sample. That's a major problem if you don't know what's in the sample to begin with, right? And we'll see examples of that in a second. When we use an orthogonal technique to measure this sample or do the same experiment, so with MRPS, MRPS measures the individual components accurately, just the way NTA did. But this time when we mix them together, we get the superposition of monomodal distributions, which is exactly what you'd expect when you mix those particles physically together. So this is just an example with polystyrene beads. How does this impact real-world measurements? So let's look at an extracellular vesicle example here where MRPS delivers more accurate size and concentration. We validate with a third technique. Incidentally, most of the stuff I'm talking about this webinar, you don't have to take my word for it. There's plenty of published literature about these differences between the technologies. And one paper I'd like to highlight specifically is this one in the Journal of EVs, especially if you're in the EV space. This is from Johns Hopkins University, and they characterize EVs and with 4 different orthogonal single particle measurement techniques. It's a nice summary of the different emerging technologies that are available for this. So I encourage you to go and check this one out. Okay, so back to our experiment. This is a real measurement here now of extracellular vesicles by NTA. And this is a fairly typical measurement for an EV sample by NTA. There's a nice clear peak around 130 nanometers in diameter in the sample. And if you're a researcher, this is nice to look at, right? It's easy on the eyes. There's a clear peak that you can track the position of. You can measure the concentration, and maybe you can tweak your formulation and see if this peak moves around or not. But you're a careful measurement scientist, and so you like to look with orthogonal techniques. So let's do that. First, let me replot this exact same data on a log Y scale. Okay, so we can see all the details more carefully. So same data. Here's our same peak. It's just dampened a little bit, right? Because the log Y scale. And now let's measure this sample by MRPS. And this is what we get. So there's obviously a striking difference between the two measurements. NTA showed us a nice peak at 130, whereas Spectradyne's MRPS shows just an increasing concentration of particles to smaller and smaller diameter to the limit of detection of our technology. And so what's going on here? Well, it's consistent with what we were seeing before, but I'm skeptical. So I'm going to take it down the hall to the TEM core and measure this sample over there. And what we see is excellent agreement between TEM, which is essentially the gold standard core tool for imaging EVs. Between TEM and MRPS, the distributions agree super well. And so what's happening here is what we saw before with the polystyrene beads, that the sensitivity of NTA to small particles when the large particles are present in the sample drops off. It rolls off as the particles get smaller. And for EVs, this is a major problem because this is a critical size range for EVs, right? 50 to 200 nanometers is the bread and butter of EV size range. So this is an example where I hope you're seeing the advantages of using orthogonal measurements to understand what's really in the sample and ultimately tell us about the limit of detection of the NTA instrument, right? So if you've been looking at NTA recently, though, you might be asking, well, so what about fluorescence NTA? So in this case, we're not detecting scattered light from the particles. We're detecting the fluorescence light. And so we won't have the same scattering limitation at small diameters. Well, so what I would say is it's still just as important to measure or to understand the limits of detection of the technology. And here's why. So here's a hypothetical example showing a peak in the size distribution at around the same diameter. Okay, so if your fluorescence NTA instrument returned to you this data, would you say that there are no fluorescent particles here? Or would you say that maybe they're just too dim to be detected, by the fluorescence detector on the instrument, and where is the limit of detection of this system? So this is the important question, and I don't have an answer, but I encourage you to try and find it. So it's important for accuracy to know the limit of detection of your technique. That's what this whole talk is about, is to understand and make sure you're working within the bounds of the technology that are appropriate for the technology. So for regular NTA, it's the limit of detection for the scattered light. And in fluorescence NTA, it's the limit of detection for the fluorescence. And that starts bringing in other questions, like how quickly are my fluorophores bleaching and things like that. So for NTA, to summarize, NTA is measuring individual particles, which is great. And that works well for monodispersed samples, as we demonstrated, right? But as we also demonstrated, for polydispersed or unknown samples, this can be a problem because the small particles are obscured by the bigger ones in the sample. This just brings it back to the same method, same message, which is that it's important to measure a sample using, look with different perspectives, right, at your sample to give confidence about what's really going on in the sample. And again, you don't have to take my word for it. This is enshrined in the EV space by the MySev guidelines. These are position paper that comes out. It gets revised every few years by the International Society of EVs. And it specifically calls out that EV characterization should be performed using multiple complementary techniques, right? They're recognizing this problem and encoding it into the boilerplate for EV experiments. And this is definitely a great step in the right direction. Okay, so finally, let's talk a bit about flow cytometry. This is an emerging method for small particles, for nanoparticles. Obviously, it's been around for decades for measuring large particles. And it's an incredibly powerful technique, right? As it pushes into smaller particle sizing and detection, characterization, it comes up against some of the similar limits as these other optical techniques. But I think it holds real promise for the future of nanoparticle analysis. So let's talk about it too. It comes with a number of key strengths. It's a foundational technology for large particle and cell analysis. It's ubiquitous, well characterized. An important aspect compared to other technologies that we're not looking at today are that the particles are now analyzed in situ, right? We don't have to immobilize the particles to characterize them. And flow cytometry is compatible with the majority of standard fluorescent protocols and labeling techniques. In fact, most of them, or many of them, were developed specifically for flow cytometry, right? So this is a well-established technology. The way it works is we send particles down a flow channel again, and we illuminate them one by one as they pass through a laser beam. And then we collect the light that comes off of these particles. They're scattered light, and typically this is collected in two different dimensions. One is forward scattered light, and one is side scattered light. And then if you excite illuminate with an appropriate fluorescence excitation laser, then you can also collect fluorescence information from single particles as it goes through. And these days, flow cytometers, they're approaching spectrometers, right? There can be so many fluorescence channels on this detection path that you can almost get spectroscopic information about particles individually. It's really incredible, especially obviously for larger particles. Now, you can ask a question, so how do we, what method do we use to detect the particle here? And there are essentially two options. One is to detect a pulse of scattered light, either in forward or side scatter channels, okay? Or you can detect a pulse in one of the fluorescence channels in order to say, oh, there's the particle, let's collect that data, that's an event, right? So when we're counting, so let's talk about where this starts to break down for smaller particles. When we're counting particles by scatter, we've already talked about how weakly the small particles scatter light, and the EVs are compounded because they're very low index contrast with the surrounding medium, so they scatter very weakly compared to other particles, especially fluorescent, especially polystyrene. And so effectively, there are definitely exceptions to this, but effectively, many flow cytometers' limited detection for EVs is in the few hundred nanometer diameter size range. And so that's really limiting at this point for using these for EV characterization in the smaller size ranges. Of course, all the big flow cytometer companies are working to drive into the smaller size ranges, so you won't have to wait long. The other option is to count particles by fluorescence, because just similar to fluorescence NTA, right? If you're not counting on the scattered light to be your detection signal, then you can count smaller particles as long as they're bright enough, right? But then you start coming up with steric limitations. So, how many fluorophores can you pack onto a 50 nanometer particle with a giant antibody on the side? It's really not that many. So the particles they get dim, smaller anyway. What about measuring the size? So we talked about counting particles, that's about concentration. When we measure size, remember that's a critical parameter also. Measuring size by fluorescence is not very well defined, right? Because a very small particle, like a fluorophore aggregate, for example, might shine as brightly as a larger dim particle, in the fluorescence channel. And so it's not a well-defined conversion from fluorescence amplitude to particle size, unless you have a specialized control assay like a control membrane dye that you make assumptions about how it's labeling other things. And that's possible. There's work going on in that area that is promising too. And then, so what about using the scattering intensity as a way of measuring the size? Just like, because we know the amount of scattered light depends on the size of the particle, right? The challenge here is to get from the scattering intensity of a single particle to the size of the particle requires the index of refraction of the particle, the refractive index. And that is a complicated question. So as an example, I'd like to just take this plot that I pulled out of the literature. This is an experiment that is showing, it's from a paper from Beckman-Coulter, which is a pioneer in flow cytometry development and technology. They have great tools. This paper is talking about how to calculate the index of refraction from the scatter signals and calibrations, right? And so what this plot is showing us is the side scatter intensity, so the intensity of the scattered light that comes off individual particles as a function of particle size, and it's known here what the particle sizes that were put in, right? So these green dots are polystyrene beads of different diameters, and since polystyrene has the same index of refraction, all these particles lie on the same index of refraction curve shown in these gray lines, right? Silica particles, these are shown in blue, they also follow a similar index of refraction curve. And then they also measured some biological particles in here too. So these show that that the side scatter depends on the particle size and also the index of refraction. So what I'd like to highlight here is if we look at trying to use side scatter to infer the particle size, we have a problem, right? It's fine if you know the index of refraction of the particle, then you can say how big it is. But if you don't know, or if your sample is a heterogeneous, optically heterogeneous sample, then if we measured 1 × 10⁵ arbitrary units of side scatter, we wouldn't know a priori if we're measuring a 100 nanometer polystyrene bead or human simplex virus, herpes simplex HSV, or a silica particle that's 200 nanometers in diameter, right? And we need some other method in order to understand what these particles are. And just to highlight the complexity of this situation, this paper was published in 2019 and is part of a very hot conversation between another research group that published a response to this paper about their interpretation of the refractive index conclusions in this first paper, and then a rebuttal to the response. And these are from just last December. So this is a very complex topic involving all kinds of electromagnetic modeling and stuff, and modeling of the particles. And this is not an easy task. Okay. So to summarize flow cytometry, I think it's got a lot of promise, and I'm looking forward to seeing the next developments in that field. We can measure size by scatter or fluorescence, right? And this works great for optically homogeneous samples. But when we start mixing them together, it's hard to get size information from just the scatter or the fluorescence, and we have to get creative with other techniques. Essentially, it comes down to using other techniques required to measure the actual size and concentration. All right, so I'd like to conclude by summarizing the 3 main points that I hope you can take away from this webinar. First of all, that accurate size and concentration are critical for good science. Remember, they are experimental parameters that must be controlled in order to perform well-controlled science, right? And they're critically related to the dose and measuring the efficacy of your product. Second, I hope I've gotten you to think about the question of the limitations of your technologies, right? So it's important to know that you're operating your technology within its well-defined, its good bounds of operation, and asking information from it that it can deliver the answer to. Finally, I hope you conclude that you can learn a lot from a different perspective, in particular using an orthogonal particle measurement technique. So thanks for your attention. If you'd like to learn more from Spectradyne, please visit our website. We have many applications listed there, information about our products, and a large and growing library of publications, videos, and applications notes. We also have a pretty active blog, including measurements of COVID vaccines that are these LNP nanoparticle therapeutics. And if you're interested in learning more about how our products can help your science, we encourage you to get in touch with us, and we'd be happy to measure one of your samples for free to show you the kind of advantages, kind of information that our technology can provide to your science. So with that, I'll say thank you very much, and be happy to answer any questions.
Thank you very much, Jean-Luc, for a great talk. Ladies and gentlemen, there are a number of questions. So what we'll do is, to make this as interactive as possible, we'll unmute those of you that have asked questions, and you can then ask the question in real time to Jean-Luc and promote discussion. So the first will be Jake Martin. Jake will unmute you, so that way you can ask your question in real time. Okay. There we go. Go ahead, Jake.
Hi, can you hear me?
Yes, we can.
Hi, Jake.
Okay, hi. Yeah, so I've recently become aware, well, thank you for the talk. I've recently become aware of another RPS-based company that advertises the ability to measure individual particle zeta potential, and I'm wondering if you can do the same with the NCS1.
Good question. So I think I know the company you're talking about. Okay, so short answer is that we don't have a well-defined method for doing that, but we'd be interested in obtaining particles to try and do experiments like that. In our experience, there's too much variability in terms of this method. There are too many parameters, and it's just kind of cumbersome. I think the idea is basically that if you throw a charged particle through with a different zeta potential, it will affect the transit time of the particle. It'll either go faster or slower. But in our experience, the logistics of doing these kind of measurements with RPS generally are rather cumbersome. So we haven't pushed too hard on that. But I'd be interested in hearing from you if you have a specific question, or if there's an experiment we could do with you about it. That'd be interesting.
Yeah, I'd be willing to try it if we can do it.
Okay, yeah, well, so shoot us an email. We'd love to hear from you.
Thank you very much. Ladies and gentlemen, the next question is by Chris Haycock. So we'll enable you to talk and you can ask your question in real time. Chris.
Awesome. Thanks again for the talk, Jean-Luc. Just curious, there's a variety of measures or measurement techniques to measure EV samples, and I was wondering how hydration of the sample affects EV size measurements when you're thinking about sample prep for something like TEM versus MRPS or other methods such as NTA. Does dehydrating the sample affect the outcome of your size measurement in these different measurements, and what your thoughts are on that?
Good question. So I'm definitely not an EM expert. Okay, the EM measurements that I showed were done by experts, and the data was provided to us. So it's my understanding, though, that cryo-EM kind of gets around that because you don't have to dehydrate the sample to measure it. So I think that's the relevant answer to the data that I showed there. In terms of how hydration affects particle size on our system, on the MRPS system, if basically anything that changes the volume of the particle will affect our measured size of it, right? Because we're measuring the volume of the particle. So if you can make the particle swell by changing the osmotic pressure or something like that, then we should be able to detect that. Does that answer your question?
Thank you. Yeah, thank you.
Okay, cool.
Thank you. The next question is by Nazar Filanov. So Nazar, you can go ahead and ask your question now in real time.
Hello, everyone. Thank you, Jean-Luc, for your presentation. It's really helpful to have some summary of different available techniques. And I have a few questions, but mostly it would be helpful, first of all, to clarify on the shown Spectradyne sample standards analysis. There are, on some of them, they present in a peak around 65 nanometers, where the graph starts. So what are they for the standards that are not 65, that peak is still there?
I'm not sure I understand your question exactly. So you're talking about real data that you've measured on the NCS1, and you're asking about a peak right near the noise floor of the cartridge, is that right?
No, I'm asking about the pictures you were included in your presentation today. Some of them have the peaks at 65 nanometers, right? And I'm just curious for the standards that, right, so here is hard to say. Can you go back?
Yep.
Further back?
Oh, further back, yep, sure. Here.
Right, so here you see some of them starts at 65, whereas basically the zero of distribution. So what are those, why they are there?
Like, what are these particles?
Yeah.
Okay, yeah, good question. So I mean, 65 nanometers would be the electrical noise floor of this cartridge size. This particular cartridge size spans from about 65 nanometers to about 400 nanometers in diameter. And when you get close to the noise floor, you will start getting some false positives detected there. So here, it's not affecting the data in any way because we're measuring the particles directly. We have features in our software that allow you to do a careful analysis very close to the noise floor and customize the acceptance criteria for whether particles are real or not down there. Does that answer your question?
It does. Thank you so much. So just to clarify, to study a complex polydispersed sample or unknown sample, how many different ranges should be exploited to just make sure you're not missing smaller stuff, bigger stuff?
Good question. So most of our customers, when they first get a system, they get a broad range of cartridge sizes. So currently it takes 3 cartridge sizes to span from 50 nanometers all the way to 10 microns, I think is right. But most of our customers are really only interested in a certain sub-range of that. And so then they, depending on their application, select the size of interest, size range of interest. So they're doing a single measurement every time. Does that make sense?
It does, yes. So for many EVs, especially with the TEM studies, people report that there is a fraction starting 50 nanometers. So do you think for EVs, optimal range will be something different than 400, maybe the one cartridge before that range?
Yeah, we offer a cartridge that goes from 50 to 300 nanometers. That's pretty optimal for EVs. I would say most of our customers don't care too much about the extra 15 nanometers though that gets you. So, you know, in all honesty, most of the samples, EV samples, look just like this one. So they have a broad size distribution that keeps going. So where you cut off here, you don't lose that much information as long as you're very careful about knowing where you're cutting off, right? That's the difficulty with the NTA method here, is that you don't know where you can stop trusting the data, or start trusting the data. Does that answer your question?
It does. Just one more little question about
Sure.
the cartridges. I know in past there was a recommendation to mix your sample with BSA or Twin. Is it our current sample prep that is recommended for EVs, or what's the best practice?
Yeah, so we have a few different options, but definitely some kind of wetting agent is required in the sample. A lot of times the sample itself contains enough background proteins or other wetting agents in there that the sample enters the cartridge on its own. But when an additional surfactant is required, our top choice would be Tween, or another one called Poloxamer, or I think it goes by Plurionic trade name, or BSA is also, also an option still.
Okay, so it's better to test the sample, and if they are concerned, to repeat with some agent added to the mixture with sample?
Yep, you can, and there, we can point you to places in the literature that have done specific studies for the lability of EVs in different surfactants. I'd be happy to help with that.
Okay, thank you. Yeah, that's helpful.
Sure.
Just one more question about sorry, am I running out?
Yeah, just a quick question, because we have a few more we want to get through. But go ahead, please.
Yeah, if I'm going too long, it's fine to continue with other questions you have, but just I couldn't catch on NTA presentation. Why is it limiting the technique that it's scattering light is different for larger and smaller particles, as that technique is measuring how particles are moving and the Brownian motion, not the intensity of that scattered light from the particles?
Yeah, that's a really good point. So the reason that it loses the smaller particles is just because it's fundamentally a dynamic range limitation of the camera. So these particles, the big ones, are so much brighter than the smaller ones that you can't take a camera image that shows both of them. So you can't see them to track them. Does that make sense?
Yeah, I would agree if we only looking at the big ones, right? But if you can see the small ones, obviously bigger ones still must be there, right?
The big ones are there, yeah, but you can't see the small ones. I'd be happy to talk, I can talk forever about this stuff. So please reach out to us and let's have a conversation about it. That'd be great.
Sounds good. Thank you so much for your answers.
Thank you.
Thank you very much. The next set of questions is from Vades Nandu. Vades, please go ahead. Thank you.
Hi.
Hi.
Yes, go ahead. Thank you.
I was wondering a way to catch the particles that are less than 50 nanometer using, the cartridges we have so far allow us to measure from 50 to 400 nanometers. So that is actually excluding a large part of your vesicles, right? Because we have some vesicles that are actually less than 50 nanometers. So I was wondering if you guys are planning to have any cartridge that can actually allow us to catch very tiny vesicles.
That's a great question. I can tell you that cartridge development and the technologies required to scale down are a very high active area for us right now. And so in the past, we have shown measurements down to 35 nanometers. Those cartridges were not commercially viable, but definitely shows proof of principle. So that's an active area that we want to expand into.
Oh, thank you.
Yep, thank you. Thanks for coming.
Thank you very much. There's a question that came in the chat box from Isabel Foreman Ortiz. So Isabel will unmute you, and you're welcome to ask your question in real time to Dr. Fraikin.
Hi, so my question was kind of a practical user question. I'm wondering if you are doing something that's time limited, like let's say aggregation of proteins. How does that work with the cartridges? How long does the measurement take? And can you keep the same cartridge in and measure that over multiple time points?
Good question. So a lot of our customers use it to characterize protein aggregation, but not usually the dynamics of it. Usually they will, they have a protein formulation, for example, biologic, and they will stress the protein to different times or different limits, and then measure the particle concentration of aggregates at different time points after that. In principle, I don't see why you couldn't monitor the concentration over time. And our technology allows you to run a single cartridge for, I think our longest measurements are something like an hour. So that's not very common, but it can be done. So certainly over the time span of a few minutes, you could monitor a change in concentration versus size over time and sort of segment the analysis that way. Does that answer your question?
Sort of. I'm also wondering for longer things like that, like if it's multiple hours, you have to change the cartridge. And what's the limitation of the cartridge? Is it drying the material out over a certain amount of time?
Yeah, good question. So we don't typically recommend reusing cartridges because they're inherently disposable, and they have features in there that can be irreversibly built up on, right, like surfaces and stuff. So we would normally recommend running a separate cartridge for each one of those measurements. And if you're concerned about variability from cartridge to cartridge, what you can do is spike in a known particle size and eliminate that completely, right? So if you spike in a polystyrene bead, for example, that is of a known size and concentration, you can normalize all your data to that, and you end up with a built-in positive control in every measurement that way to eliminate any kind of variability from different cartridges. Does that make sense?
Yes, thank you.
Okay, thanks.
Thank you very much. There's a question that came from the chat box from Mandy Chan. Mandy, we're going to unmute you so you can ask your question in real time. Thank you. Mandy, you can go ahead and unmute and ask your question. Okay. Right. So I guess you can ask your question again in the Q&A, and we'll ask Dr. Fraikin to respond. Another question came in from Daniela Stadler. Daniela, we're going to unmute you, and you can ask your question in real time. Thank you. Hi, Daniela. Would you like to ask your question? Okay.
Yes, thank you. Can you hear me now?
Yes, we can. Go ahead, please.
Okay, so I'm sorry for that. Thank you for the webinar. So I wondered if I would like to measure EVs with a particle size of 100 nanometer, which cartridge size would you recommend? So is it better to go with C300 or the 400 cartridge?
If it's really 100 nanometers, I would recommend the C400s. They're just more forgiving from a perspective of the wetting agents and things like that. So they're just generally easier, but either one will work.
Okay, thank you. And can I have a follow-up question? So I expect a lot of large particles, and I read that there is inlet filters in the cartridges. And I wondered which size is this inlet filter and how I should prepare my samples beforehand. So meaning if I pre-filter them, which filter size do I use?
Great question. So one of the details of our technology we didn't get into is that on every analysis cartridge, there's a built-in pre-filter that excludes particles that are larger than the nanoconstriction, right? So that prevents frequent blockages of the constriction and enables you to run very complex samples, including those that have bigger particles, much more easily. So my first response would be to try without pre-filtering and just rely on the cartridge technology to do that for you. The filter cutoff for like a C400 cartridge would be about 400 nanometers and would stop anything bigger than that from going through. Does that make sense?
Yes, thank you very much.
Okay, thank you.
Thank you. So there's a few questions which Dr. Freykin can respond to offline because they're very specific to the particular user. We're going to unmute a few more people so that they can ask their questions. The next one is Mikhail Spanos, and he asks a question. Please go ahead, Mikhail.
Hello.
Hi.
My main question is, so the precision medicine is focusing on smaller size particles. And the question is how Spectradyne is going to tackle with the particles size less than 50 nanometers, and if you're going to develop any cartridges for that in the future.
Good question. The answer is yes, and it's an active area of development right now. As I mentioned, I think we've demonstrated some measurements successfully down to about 35 nanometers, so we know it's possible. It's just not ready for commercial prime time yet.
Thank you so much.
Thank you.
Thank you very much. We have a lot more actually questions, so we'll take a few select questions and then we'll defer the rest for an offline response. The next one is by Samira Saif. We're going to unmute you and please ask your question for Dr. Freykin. Go ahead.
Hey, can you hear me?
Yep. Yes, we can. Go ahead.
First of all, thanks for the nice presentation. That was very helpful. I was wondering if you have ever, measured polydispenser particles and compare your data with, from NCS1 versus TRPS.
Oh yeah. So I think the primary. So we have not done a direct comparison like that. I believe others in the literature have. Fundamentally, our measurement, microfluidic resistive pulse sensing, works on the same principle as tunable resistive pulse sensing. It's really just a matter of ease of use and practical utility. So our technology is very fast and easy to use. We use single-use disposables, so you don't have to clean anything. And because of the built-in pre-filters in the cartridges, which is our patented technology, by the way, that enables the analysis of much more complex samples, than the sort of simplistic TRPS implementation. Does that make sense?
Yeah.
Okay.
Yep. Thank you so much.
Thank you.
Thank you very much. We're going to take one further question, and that's from Christina Sandu Aparicio. Christina, please go ahead. You can ask your question. Thank you.
Hello, can you hear me?
Yes, we can.
Hi, thank you so much. So we are assuming that a lot of our particles are more sheet-like or rod-like in nature. And what are the limitations of measuring our particles on the Spectradyne? Would we have to maybe just do multiple cartridges? Because again, if the pre-filter does not filter out those larger particles, if they're going in long ways, I would assume that if we're using a cartridge size that is smaller than that length, then that data would not show up, right? So it essentially might look like it didn't get picked up at all. So is the best way to go around that is using multiple cartridges to kind of filter out what we can, or would it just be very biased? And I guess.
Sorry, I don't want to interrupt. That's a great question, and I think what ultimately maybe what you're asking for is how does shape affect the particle measurement, right? And are there any practical considerations for measuring oddly shaped particles? Is that accurate
Yes.
assessment? Okay, so it's a great question. Fundamentally, our technology measures the volume of the particle and reports an effective spherical diameter. And so our limit of detection is an effective spherical diameter. It's, speaking very carefully, it's the volume of a particle whose effective spherical diameter is 50 nanometers. So if you have a very thin sheet, it's going to have to be huge in order to add up to enough volume to meet the volume of a 50 nanometer particle. However, we have many examples of oblong particles, especially weird shaped viruses and stuff. And I know we call a couple of those out on our blog page. So if you have a chance to go and check those out, there are some discussion about how shape affects the measurement over there. Practically speaking, when people need to measure a very broad size range of particles, they start with the biggest cartridges, figure out what's in that size range, and then go to the smaller ones. And the data automatically stitches together essentially. So you can piece together the full size range that way. Does that answer your question?
Yeah, that makes a lot of sense. Thank you so much.
Okay, thanks, Christina.
So at this point, we'd like to thank Dr. Freken for a great talk. Ladies and gentlemen, as you can see, so much engagement. And so we'd like to thank all the participants, all the people who asked questions. There's a number of other questions that Dr. Freken will respond to offline to each of you. And we'd like to thank you once again for your participation, and thank Spectradyne once again for a great webinar as always. Thank you, Jean-Luc, and we look forward to seeing you at the next Spectradyne webinar. Thank you and goodbye.
Thank you.